Now I have the pleasure of introducing Dr. William Chronister. Will be presenting T-Cell Tool Spotlight, TCRMatch. This is a really great new tool that was released earlier this year, which we'll delve into a little bit more. And we're just about 3:45, Will, so I'll let you share your screen and kick us off. Thank you.
Great, thanks Nina, and thanks to all the attendees. Hope that this workshop has been useful for you so far. Today I'm going to be presenting on TCRMatch. It's one of the newest tools that we've added to the IEDB analysis resource. And we published this earlier this year, if you're interested in reading the paper. Like I said, it's available on the analysis resource, and it's also downloadable as a command line tool if you prefer to run it that way. So just a brief bit of background. We've covered this already today, but the focus for TCRMatch is on the recognition of antigens or epitopes by T-cell receptors. We have a lot of tools that focus on sort of the left half of this diagram, which is the presentation of antigens by MHC. TCRMatch focuses on the right half of this, specifically the interaction between the T-cell receptor and the antigen. So the T-cell receptor, as you may know, is a dimeric protein complex that consists of a beta chain and an alpha chain. And these chains are comprised of VDJ gene segments that are recombined in diverse ways that generate the vast diversity of TCRs that we have in our immune system. And each of these chains contains 3 CDRs or complementarity determining regions, numbered 1, 2, and 3 for both alpha and the beta chain. One of the techniques that has become very common in T-cell biology is repertoire sequencing. This is a form of sequencing that allows you to produce the TCR chain sequences that you have within a population of T cells. And in particular, a lot of these approaches focus on the CDR3 beta region, which is generally considered to be the most important for T-cell specificity. And this is because it's the portion that interacts most directly with the epitope. And it's also the most variable region of all the CDRs. As you can see, let me switch to my laser pointer. The CDR3 beta region spans from the V gene segment through the D and J segments. And for that reason, you get a lot of diversity in that small region. And it is relatively small. These are some example sequences of CDR3 beta of that region. If you're not familiar with them, this sequence is bounded by a cysteine at the N-terminal end and a phenylalanine or sometimes a tryptophan on the C-terminal end. And so the portion in the middle is the CDR3 beta. Now, one thing about TCR sequencing is that it doesn't directly reveal the epitope that's recognized by a given CDR3 beta. You can do other experiments to help you find that, but on its own, it doesn't tell you anything about what the epitope is. But one thing that TCRMatch is intended to do is to use known CDR3 beta epitope pairs to make predictions for CDR3 beta sequences that you might have from a sequencing run like this. So again, our goal was to build a tool that can search input CDR3 betas against the IEDB, where we have a bunch of known sequences and known epitopes. And using the principle that similar CDR3 beta sequences often recognize the same epitope, we can then generate informed, educated guesses or candidates for what the epitopes might be for your input sequences. So to do this, we gathered a data set from the IEDB consisting of 24,000 or so CDR3 beta sequences, as well as the 1 or more epitopes they were reported to be specific for. And then we tested 7 different similarity algorithms because we had to figure out, well, if you have 2 SCARE3 beta sequences, what's the best way to determine which ones are similar to each other? And there's different ways you can imagine approaching that. So we had 4 alignment-based methods. We tested Levenstein distance, which is the number of edits you need to make to transform one sequence into the other. We tested TCR dist, which is another distance-like metric. And then we tested TCRMatch, the algorithm that came to be known as the TCRMatch score. This was previously implemented by or reported on by a couple of other papers. And I'm going to spend a little time delving into what this algorithm does. So the algorithm relies upon this BLOSUM62 scoring matrix or similarity matrix that has been around for around 30 years now. It's very common in alignment of sequences. But basically, this comes from, if you don't know, it comes from blocks of evolutionarily related proteins. And basically, the creators looked at how often certain residues were substituted for other residues. And depending on how frequently that occurred or didn't occur, you could generate a scoring system for how favorable or unfavorable a substitution is for one amino acid to be replaced by another. If you intend to conserve the functionality of that protein as the protein is changing its sequence through evolution. So to give you an example, you can look at amino acids like cysteine or tryptophan, and they have very high positive scores. So cysteine to cysteine is a 9, tryptophan to tryptophan is 11, and that's because these are very particularly structured amino acids that have very important roles to play, and you can't just swap them out with any other amino acid. It'll radically change the shape of the protein. On the other hand, things like leucine and isoleucine here in the middle, these all have positive scores when they're swapped for each other because they're very similar in structure. And so that gives you a little bit of an idea about how this scoring system or this scoring matrix can be used to determine how similar 2 sequences are to each other based on how similar the amino acids are to each other. So the algorithm that TCRMatch uses compares 2 sequences using all possible k-mers, and it's going to do so using this BLOSUM62 matrix. So it'll start by looking at each amino acid individually. So we start with k equal to 1, and we compare A to A, checking the score for how good that comparison is, and then it compares A to S again, A to I, and so on for k equals 1. And it'll go through every single possible combination between the 2 sequences. And then it'll switch to k equals 2. And so we'll look at each dimer, and within each dimer, we'll look at the first in the pair, and then we'll look at the second. So A to A, S to S, A to S, S to S, and so on down the line. And it'll continue doing that basic procedure for k equals 3 all the way up to k equals 10, which is the length of this shorter sequence. And ultimately, it uses all of these comparisons to generate a score between 0 and 1, where a perfect match, the exact same sequence, would be equal to 1. To evaluate how well this algorithm performs, we ran our data set against, we ran each sequence in our data set against the rest of the data set, and we calculated TCRMatch scores for each comparison. If we impose a threshold for a match, if we just set it at 0.90, that will eliminate this 4th potential match, and we're left with 3 scores that exceed 0.90. And then we can evaluate how well we did with this threshold by looking at the epitopes that are recognized by the input sequence and seeing how that compares to those recognized by the rest of the data. So we can see that epitope 1 is found in the first sequence and the third sequence. epitopes 2 and 3 are not recovered by any of these matches. And then these matches additionally recognize epitopes that are not recognized by the input sequence. And so altogether, we can take these false negatives and false positives, and we can turn this into a precision recall plot, which is what we did for TCRMatch as well as the other metrics that we tested. And you can see TCRMatch is here in pink, and it generally performs pretty well by eye. We noticed that we also did a random model where we essentially shuffled the CDR3 and epitope pairs, and we found that just choosing finding a match by random leads to performance that's right along this dashed line here. So we were pleased to see that all of our methods did perform better than a random model. We also we wanted to verify that TCRMatch was indeed the best metric, so we ran several resamplings of the data set. We used a bootstrapping approach to look at how TCRMatch fared over 100 resamplings compared to some of the other contending methods. And we found that with a p-value of less than 0.01, it was significantly better than the other metrics on this data set. We also ran all of the metrics on a separate data set published by 10x Genomics, which featured about 3,000 sequences and 18 epitopes. And once again, we saw TCRMatch be the superior metric across the precision recall plot. We also had, within this data set, we had 232 CDR3 betas that had exact matches in the IEDB, of which about 73.7% recognized the same epitope that we had in the IEDB. So that gave us confidence that, again, these epitopes are definitely predictable with the right, when it comes to similarity, we can always, we have a good idea of what, of how to predict epitopes based on the sequence of the CDR3 beta. So I'm going to quickly demonstrate how this tool works. It's pretty simple to run, I think. So this is on the tools website. We have the TCRMatch tool page here. We have a big space where you can enter in manually or copy paste in your CDR3 beta sequences. You can also upload a file if you so choose. I'm going to run the example data, which is available in this example tab. And I'm going to choose the slightly shorter one. So I have 3 sequences here. And then we just have a couple of options that you can use to customize your analysis. Trim sequences refers to trimming off that, that cysteine or phenylalanine residue that might be at the beginning and end of your sequence. So just to give you an example of how this works, I'll go ahead and add those on. We recommend you keep this checked just to ensure that we can find matches for your data set if you do have the C and F residues included in your CDR3 beta sequences, which sometimes some softwares include. If you don't have those C and F residues, you don't need to worry. It's not going to trim off anything from the actual CDR3 beta sequence. You can choose a filtering level. You can choose to retain matches that score above 0.84, 0.90, or our recommended threshold is 0.97, which is pretty stringent, but you will get more high confidence matches that way. And you can also choose to show all matches if that's preferable. I'm going to leave it at 0.97. And then here you can enter your email address if you want your results to be emailed to you, if you're submitting a large list of sequences. In this case, this should run pretty fast. It's only 3 sequences. Okay, and so you can see the results are here, and basically we have the input sequence, we have the match that was found within the IEDB, we have receptor IDs that you can click on to go and explore more information about the data that was entered, was curated by the IEDB about these sequences. You can see the epitope candidates that are listed here. So we have one for SARS-CoV-2. And then we have 3 for human herpes virus 5, which is, I believe, where these sequences came from. So you can click on any of these receptor IDs and follow this to get more information about, again, where the data came from. If you want to look at the publication it was sourced from, then that's available as well. You can follow this through and filter the IEDB in different ways to learn more about the data set from the paper, whatever you want to do with that. There's lots of possibilities. So I will now switch back to my slides, and we do want to implement some changes to the output. You might have noticed it's a little bit cluttered. This is what the new output is going to look like when we update it. It's going to keep it a little bit more simple and put the receptor IDs on separate lines. And so you can look forward to that in the near future. So in summary, TCRMatch is an efficient way to find similar TCR sequences that have been curated in the IEDB that have a known specificity. You could use it with other TCR clustering tools like LIFT2, if you wanted to sort of cluster your TCRs first and then run each cluster through TCRMatch. That might help you find and make sense of the potential epitopes that they might recognize. One thing that we like about TCRMatch is that, unlike other tools, it's not trained on specific epitopes, which means it's unbiased and it's generalizable. So it doesn't require a huge data set to be able to find an epitope candidate. All that it really requires to find the right epitope is that the epitope has been studied and has been curated within the IEDB when you run the tool. So it won't be able to find everything, but if it's in the IEDB, then at least there's a chance that you'll find the right matching epitope. In the future, we're going to keep adding to the IEDB, obviously, and we believe that'll only improve the quality of TCRMatch. We also have some other improvements in mind, such as including paired analysis if you have CDR3 alpha chain sequences as well as CDR3 beta. That should help improve our performance. We're also going to add MHC data to the output to give you a little more information about each match. And finally, we're going to be adding enrichment scores in the output, which should help boost the signal to noise ratio, because we do occasionally get false positives that show up in the results due to the large amount of data that's been deposited in the IEDB for specific epitopes. So with that, I want to thank everyone that was involved in creating this tool and writing the paper, and especially my co-first author, Austin. And the links to TCRMatch are here if you're interested in trying it out. So with that, thanks for your time, and I'll pass it back to Nina.